That was purely by what we call serendipity. So just luck, pure luck. So when I was in my third year at university, we had to choose a project to do for 10 weeks, an experimental project in one of the labs. And this was the Department of Biochemistry in Cambridge. And we had presentations from the different lecturers. And one of the lecturers was Tim Hunt. And he was the most interesting of all of them. Not really for the project, but because he was the most dynamic and exciting of the lecturers that we had. He was completely different from everybody else. So a lot of us really wanted to do a project with him, and luckily I was one of them. So I think in the end he took 5 people because he really likes to teach young people. And at the end of my project, Tim said to me, would you like to stay on and do a PhD? So I said yes. And when I started my PhD, he gave me a choice because the lab worked on protein synthesis, but he discovered cyclin the summer before. And so he said, you can either work on protein synthesis or you can work on this protein that I've just identified. And I said, I'd like to work on the protein he just identified. So just pure luck. Absolutely nothing more than that. So pretty much when I started my PhD, all we knew about cyclins was that they were proteins that were synthesized after you fertilize a sea urchin or a clam, and they increased and then disappeared. They increased and disappeared. That was it. That's all we knew. And so pretty much during my PhD, Tim and I learnt molecular biology together because he was a biochemist. So we had to learn molecular biology and then clone cyclin. And it was only really at the end of that, when we had the clone, that we could really begin to understand what it did. Until then, we had lots of complicated models for how cyclins might connect with the cell cycle, but nobody in the cell cycle field, which at that time was dominated by guys working on yeast and guys working on xenophus, no one in the field really took cyclins seriously. There was the idea that there were maybe just some protein that was made by sea urchins and clams. So it was really Tim who managed to talk to all these guys and convince them that there was something interesting there. And finally, when we had the clone, we could show that it was directly involved and not some sort of extraneous player in the cycle, but actually something really central to it. Yeah. Well, cloning human cyclin B was much easier than cloning sea urchin cyclin B, because sea urchin, we had no other clue to it than just the protein size and that it was made in sea urchins. Once we'd got the sea urchin, then we were able to get the xenophus, and then if you compared the sequences, you find these conserved bits, the cyclin box. So when I went to do my postdoc supervisor, Tony Hunter, he was interested in any sort of anything to do with cell growth and particularly kinases. And so he said, why don't you try and clone human cyclins? So I just used the conserved sequences and found cyclin B actually quite quickly when I started my postdoc. And then I had to learn how to use human cells, how to synchronize them, how to study their division. So then, actually, within about a year and a half or so, we had all the data for the first paper. So that, I think, is the fastest that I've ever managed to do a project. The impact, I think, it was our cloning of human cyclin B that made people think about cyclins as being important for all division. So not just invertebrates or xenopus, but everything. And very quickly, people identified other types of cyclins through the conserved cyclin box. And one of those, the D-type cyclins, they are very strongly connected to disease, to cancer. They get amplified in a lot of cancers. And so the cancer field suddenly took an interest. And that exploded. So it went from being, just the small cell cycle field to the huge cancer field. So I think that's really why that paper is important. It was a step towards the connection between cyclins and cancer. So that was all those foundations were there, and very important work was done to find out how the APC and the spindle assembly checkpoint interacted. So my contribution was to develop this assay to look at the degradation of proteins in living cells. And that was because we got interested in using green fluorescent protein to look at where proteins are in the cell. So that came from my work with Tony Hunter, where we were doing immunofluorescence. But the problem with immunofluorescence is you have to kill the cells to look at the immunofluorescence. So you can't see what that cell just did, and you can't see what it was just about to do. So I always wanted a way to be able to look at changes through time. And so when GFP was identified, I thought we could use it to tag proteins and see where they go. So we were doing that, and then again, serendipity, the cyclins, when you tag them, the GFP shows where they are in the cell, but it also, the signal disappears when the cyclin gets degraded. So that turned out to be an assay not just for where proteins are in the cell, but also for their stability. And we identified that cyclins get degraded very quickly at one specific time in the cell cycle. And so we could start to use that assay to look at their degradation. And then that led on to the APC and to the spindle checkpoint. It's begun to change because I'm surrounded by people who work on cancer as a disease and not as a sort of theoretical concept. So I have colleagues who are treating patients in the clinic, and then they have their own research labs. So talking to them makes me understand how complicated cancer really is, and it's not a disease that you can just study in a dish. You really have to have a lot of sophisticated models for cancer to be able to understand it. And so what we're beginning to do now is try to see how our knowledge of mitosis and cell division can help my clinical colleagues in terms of designing better drugs to treat people. And then they're helping me to understand how cancer really evolves over time and how the different cell populations interact to change the behavior of the cells and change the behavior of the tissues. So slowly, we're beginning to develop ideas about how we can use drugs that affect mitosis to begin to treat particular types of cancers. So I think what's interesting about that, what we discovered in that paper, is that the cyclin B CDK that triggers mitosis, we've known for a long time that it reorganizes the cell to make the mitotic apparatus and to coordinate changes in the cell. And what we found is that in altering the cell architecture, it not only sets up the apparatus to divide the cell, but it also sets up, or is important to set up, the regulatory machinery that prevents the cell from dividing too early. So it both triggers the machinery that will eventually divide the chromosomes, but at the same time, it sets up the regulation to stop that from happening too soon. So it's a beautiful example of how the cell coordinates things in space and time. I must admit, I didn't really expect our research to have much impact on science. So I've been lucky in that some of the things that I've been involved in turned out to have been effectively sort of the right place at the right time. So the early work with the cell cycle, and then later work with green fluorescent protein. So those had an influence simply because I happened to be working in those areas at that time. So I think now probably the major impact we've had is partly the identifying the human cyclins and their regulation, partly using GFP as a live cell assay for proteolysis, which a lot of people now do, and then a little bit in terms of how the spindle assembly checkpoint works. But I must admit, that's really been building on all this other people's work for 20 years before we started on it. So those are probably the areas that I've had the most influence. In terms of the future, then our own research, I think, hopefully will be able to show how you can measure the way in which proteins form complexes and the kinetics with which that happens in living cells. So that's what we're now trying to develop, to see how proteins come together in particular places, form a complex, do something, and then the complex disassembles. And we're doing that in living cells. So we're trying to do in vivo enzyme kinetics. So I hope that will have an effect.